right. Oh, can you hear me? Okay, so hi, thanks for being here. My name is Sarah Lemer. I am a research faculty at the University of Guam. And my talk today is about finding genomic signatures of evolution in Nautilus, an endangered living fossil. So the first question that should come to your mind is, what is a living fossil, right? Is that really a thing? There's different categories of organisms, actually, that fall into this living fossil descriptions. The first thing that comes to mind is the coelacanth, or the horseshoe crab, the ginkgo tree, the horsetail, and Nautilus. So what makes a living fossil organism living fossil? Well, there's a few rules that you need to abide to be part of this club. One of them is that you need to be part of a once diverse and widespread lineage. So the fossil record must show this. The second is that you must have originated millions of years ago. So Nautilus, for example, originated about 100 million years ago. The third one is that you need to show some morphological stasis. So basically, your morphology hasn't changed much when compared to the fossils. And now you should show low taxonomic diversity. For example, the ginkgo tree only has one species in this whole group. Nautilus is 6 species that are described right now. So those 6 species are spread into 2 different genera. The first one is Nautilus. It has 5 species, and if you look at the pictures here, it's very difficult to distinguish them. However, some people manage to. The 6th species is its own genus, and it's Nautilus corpiculatus, and it's easily distinguishable by this furry shell. So what else do we know about these iconic deep-sea cephalopods? Well, first, their distribution is Indo-Pacific, from Thailand to American Samoa. They have limited dispersal ability, so they usually stay on the continental shelf just because their shell will implode under 800 meters. So they go deep, but actually not that deep. They also have lower production rates, so they reach sexual maturity at about 15 years old, and their eggs take about a year to hatch. And finally, remember how I told you there's 6 species total? Well, they were all described based on shell morphology only, except for one species, Nautilus balawensis, who was found in Palau, and it's the only one that actually has soft tissue and shell used to describe it. So Nautilus are part of that living fossil club. Now, what does that mean in terms of their evolutionary history and evolutionary biology? Well, there's a few hypotheses that have been put out there about living fossil evolution. The first one is that because of their morphological stasis, we most likely are in presence of high cryptic diversity. That's an easy one. The second one is there's also expectation of low speciation. That is because there is a perceived evolutionary stasis as well. For example, for the coelacanth, a paper in 2011 in Marine Genomics called their genome evolutionarily frozen since several millions of years. The third hypothesis is that today there is no contemporary diversification in these groups because they were once part of a huge, widespread diverse group, and they are not anymore. And the last one is we noticed that in a few of these species there have very little population differentiation, so we suspect they have high effective population sizes and low genetic drift. So we wanted to test these hypotheses on the genus Nautilus and see if they were actually correct or not. So the approach we used was a double digest right-seq. At this point, I'm not going to describe what the method is. I think you all know it. We were lucky to obtain 140 samples from all over their distribution range, thanks to our collaborators, Peter Ward and Neil Langman at the AMNH. We even got some unknown specimens that were lost in the museum without label. With this data, we generated over 170 million reads sequenced on an Illumina. And this raw data, after being trimmed and demultiplexed, is available on NCBI if you want to play with it. And then we applied multiple analysis on them just to answer our different hypotheses. So for example, we assembled the loci and validated them with stacks and bow tie using a transcriptome. And well, it would have been better with a genome, but it's not available. And this data was then used to do phylogenetic inferences. So that data is actually 19,000 loci, SNPs, sorry, 19,000 SNPs that we use for phylogenetic data set. We also wanted to do some population genetic analysis, and there we wanted to make sure we avoid linkage disequilibrium because we don't know where those rad tags are located on the genome. So to do so, we only use the first SNPs of each loci. And so that reduced our data set to 2,500 SNPs. And then we applied multiple population genetic analysis that I will go through the presentation. So the first result I wanted to show you is the one with the first data set. So this is the tree that we obtained with IQ tree analysis in RAXML. And we find three main clades represented by green, blue, and orangish colors. And we also went ahead and ran a structure analysis with the 2,000 and 5,000 SNPs only. And it's right next to the tree, and you can see it actually shows the same categorization of those three clades. So we have three clades, three genetic clusters, and these are the South Pacific, which is comprises the American Samoa, Vanuatu, New Caledonia, and Fiji, so basically all the oceanic islands. Then we have what we call the Coral Sea clade that has samples from Papua New Guinea and the Great Barrier Reef. And finally, the Indo-Pacific clade, which is much wider geographically spoken clade. We have Palau, Philippines, Indonesia, and Western Australia. The next thing we notice on this tree is none of the morphospecies described are valid except for one, which is Nautilus microphalus up here is the only monophyletic clade, and it is found only in New Caledonia. All the others are spread all around the tree. So cryptic speciation, yes. Another interesting thing, we calculated FST values between our three clades, and there's no admixture at all between the clades. There's actually 35% FST values between South Pacific and Indo-Pacific. So we most likely have three species for sure. The next thing we wanted to know, it looks like there's different things happening within each clade, so we wanted to see if we have substructures, so we ran basically structure within each of those three genetic clusters. And what we found is in the South Pacific clade here, we basically have a pattern of genetic isolation where each island is its own genetic population, except for Samoa and Fiji who cluster together. In the Coral Sea clade here, you can see a pattern of admixture from Papua New Guinea to the Great Barrier Reef. And in the Indo-Pacific, there's basically ammixia and no genetic differentiation between those populations, although they are the most widespread. So it's pretty interesting that we have three different patterns in those three clades. So we wanted to look even further and see if we could explain this by different methods. So the next thing we looked at was signatures of selection. So we used different methods, and actually I forgot to show you, but there was a QR code on the first page. You can scan that, and it brings you to the paper where you can see all the different analysis we did. But the one I want to show you today is we basically calculated FST values for different comparisons between clade and populations. And we compared FST values on untranscribed, so neutral loci here on the x-axis versus transcribed loci here on the x-axis. So basically all of this up here are loci that are that have higher FST values on untranscribed loci than transcribed loci. Did you get that? Untranscribed versus transcribed loci. So what we first see is that we have higher FST values at transcribed loci than untranscribed loci, so everything above that line here. The next thing that we notice is that, for example, this point here is the comparison between South Pacific and Indo-Pacific clades with oceanic islands and the widespread clade, and we have again this pattern of higher FST values on including loci. And all these other points here are basically individual comparison between the South Pacific individual populations and all the other Nautilus populations that we have. So they're mostly all on that upper part of the graph. So what it basically means is that we have genome-wide selection pattern in all the populations of the South Pacific clade, and not the other populations. The next thing we wanted to look at is if this hypothesis whether living fossils have higher effective population sizes was true for Nautilus. So what we did is here we ran ABC simulations. We have population sizes here in the different populations. What we first notice is we have very large population sizes, about 4 million, 7 million, and 6 million effective population sizes for the Coral Sea and the Indo-Pacific Lake, but we have smaller, much smaller population sizes for the South Pacific Lake. So, however, altogether, these effective population sizes are much higher than they are for other cephalopod species. So what does all that mean? If we try to bring all of these patterns together, in the South Pacific Lake, we have small isolated oceanic islands. They show high FST values between all themselves and all other populations, and the FST values are higher on transcribed loci, which means we have, that's a strong sign for genome-wide positive selection, and that indicates somehow ongoing diversification and speciation in this South Pacific Lake here. In the Coral Sea, we have a pattern of admixture. So we have a southward migration from Papua New Guinea that is introgressing into the Great Barrier Reef. We have no genome-wide positive selection here. The FST values were equal whether they were on transcribed or untranscribed loci. Those two populations were separated for a very long time until recently, yet they are able to interbreed. So we have admixture which seems to indicate lower rates of evolution in this clade. In the Indo-Pacific clade, here we have, so it's the widest clade in range. However, most of it is on continental shelf except for Palau, which is a little, surprising. It looks like depth is a stronger barrier to dispersal than actual distance. We do have high effective population size compared to other cephalopods. So high effective population size and high migration basically mean no genetic drift. So if we go back to those 4 hypotheses that we had before, we have high cryptic speciation in Nautilus, yes. Low speciation rates in the Coral Sea clade, yes. No contemporary diversification, no. We actually have ongoing diversification and speciation in the South Pacific clade. And high effective population size and low genetic drift, yes. So we basically say yes to 3 out of the 4 evolution hypotheses for living fossils. So that was the presentation. I would like to thank all the people that participated in this project, mainly the people that sampled, Gregory Barrett, Peter Ward, Neil Landman, and research computing and Bauer Core at Harvard University that helped generate the data, and my collaborator that when we tried to go sample in Thailand, it was a total failure. We didn't get it. That's why there's 0 in Thailand. And if you scan this QR code, I will bring you to my lab webpage where you can see more of the research we're doing, and we're right now recruiting master students. We have 3-year stipends, so if you know someone who's interested, just